Hello again, IPM students. So, today is the recorded lecture for April 4th. It's a Tuesday. So if you recall, today I am in Portland at the regional meeting for the Entomological Society of America. So I have canceled lecture and lab. This recording is our substitute for the April 4th lecture. You will have to know this information for the exam. So make sure to watch this. Originally, as per the syllabus, we were supposed to cover some new ground on sort of pesticide safety, but we didn't get to the end of the managing pesticide resistance, so I decided that with today's lecture, we will just wrap up resistance, and we'll touch on pesticide safety later. So just so you know, there is no quiz today, so that's a freebie. I'll just give you 2 points. And yeah, that should be it. So just some announcements before we get going. As I mentioned earlier, there is no lab today because I'm not here. However, I will be back tomorrow on the 5th, so we will be having our regular lab on Thursday the 6th. The lab will be at Bayer Crop Science. Again, that's 266 South Monroe Avenue in Fresno. It's right off of 180. About maybe 10 minutes away from Kerman. So what you can do from campus is you can just hop the 168, take it west, then get on West 180, and you just keep following 180 until you see the intersection for Monroe Avenue. You'll turn left so that you're heading south on Monroe, and then Bayer Crop Science will be immediately on your left about maybe a quarter mile, a third of a mile down the road. It's got a great big sign. You can't really miss it. So if you're driving yourself, that's how you get there. If you're riding in the van, I will pick you up at room 119 at 2 o'clock. So with that, be at Bayer Crop Science at 2:30, ready to go. Now just a reminder that there is a specific dress required for Bayer Crop Science. This is for your own safety as well as Bayer Crop Science's liability. They want to make sure that you are wearing long sleeves on your shirt, long legs on your pants, and you need to have closed-toed shoes. So basically no exposed skin other than like your hands, your neck, and your face. That being said, you'll also have to have eye protection. So if you have sunglasses or eyeglasses that more or less wrap around your face, you'll be fine. Otherwise, when you show up, they will provide you with a set of protective lenses. In addition to these required dress, you are also recommended to have sunglasses, hats, and sunscreen just for sun protection. Again, this is recommended, not required. Alright, so last time what we really talked about was what resistance is and what are the factors that can cause resistance. And then towards the end of the lecture we got into reactive, which would be essentially no management, versus proactive, which would be basically actively trying to avoid resistance, as 2 different types of management techniques to control for resistance. What we're going to be doing today is digging into these specific management resistance tactics. So how do we actually proactively try to stop resistance from emerging in the field? Now, overall, both of these ways are really based in manipulating the natural selection that favors resistant individuals. So, just to review briefly, right, we have resistant individuals in the population. They are carrying a specific allele or a gene variant that makes them resistant, and every time we spray a pesticide, they survive, whereas we kill off the majority of the susceptible individuals in the population. So when the mating period comes around, we have a disproportionately large number of resistant individuals and a small proportion of susceptibles, so that in the next generation we have more resistant individuals than we did in the one before. So 2 major ways that we get at this problem is that first off, we can reduce the selection pressure that favors the resistance allele. So essentially we can make it so that less of the susceptibles die, so that we end up with a larger susceptible population that can outcompete the resistant ones for mating. On the flip side, say we have a technology where we can't really control the selection pressure, something like instance, BT crop, right? BT corn, BT cotton, they express BT at a high concentration all the time. And so it's not like we can reduce that selection pressure. So instead, what we would do is artificially increase the number of susceptibles that are available during the mating period in order to mask the resistance genes. So we'll dig into each of these approaches separately, and that will really be sort of the long and short of this lecture. All right, so the role of selection pressure. First off, let's talk a little bit about selection pressure. In natural selection, a selective pressure is essentially any environmental factor that favors one trait over another. So if you're a plant growing out in the desert, and every once in a while you're exposed to prolonged periods of drought, then drought would be a selective pressure that favors drought-resistant traits. In the case of pesticide resistance, selective pressure is the pesticide itself that kills the insects. It's a particular trait or pressure that favors the resistance trait. And generally, the greater the mortality that's caused by the pesticide, the greater the selective pressure is. And thus, the faster the resistance will develop. And this all basically comes down to the fact that the higher the selective pressure or the more mortality there is, the more susceptible pests will be killed, and the greater the proportion resistance will make up at the reproductive phase. So just to walk through an example of this, let's say we have a pest population, and it's got 100 individuals in it, and only 1% of the population, 1 out of every 100 is resistant. Now, admittedly, this is a pretty high resistance rate. Normally in the wild, we're talking about hundredths or thousands of a percent being resistant, whereas here we're talking about 1 in 100. But I don't want to draw 1,000 or 1,000,000 little circles, so we'll just stick with this nice little scheme here. So let's say that we apply a pesticide that has somewhat low mortality. It only kills 70% of all individuals that are susceptible to it. So when we apply a 70% mortality, we expect 70% of 99, which is close enough to 70 that I'll just keep it at 70. We expect 70 or 7 rows of these susceptibles to die, leaving us with a final population that has 30 susceptibles and 1 resistant individual. So that now about 3.2% of the population is resistant. And thus, in the next generation, we expect about 3.2% of the population to carry resistance genes as well. And this is a 3-fold increase from our original rate of 1%. So, as you can see, at this low mortality, we see an increase. And it's actually a pretty substantial increase. 3%, or yeah, a 3x increase is pretty substantial. But what if we applied a really effective insecticide or pesticide, right? Something that's going to kill a lot more susceptibles than 7 in 10. Something like 95% mortality. Instead of having 30 resistant or susceptible individuals, now we only have 5 surviving. So that we go from a 1 in 100 ratio for resistance to a 1 in 6 ratio. So resistance is now 16.6% of the population, which is approximately a 17-fold increase in the proportion of resistance. Now, admittedly, you're not going to get as many pests in the next generation because you've got less individuals, they're going to lay less eggs, you're not going to get as big of an outbreak next time. But on the flip side, those individuals you do get, a higher proportion of them are going to be resistant. So, the more pests we kill, the higher our mortality, the faster resistance evolves as compared to our low selection treatment. So consequently, many of the techniques based around controlling resistance are based around reducing the amount of selection we provide in order to increase the number of susceptible pests that are left for mating. Right? Just because having more susceptibles around is going to increase the chance that the resistant pests will mate with the susceptible ones. And in that case, the offspring will inherit both resistant and susceptible alleles, and it'll be more likely that those offspring are going to be susceptible. So it's kind of a double approach. On one hand, by increasing the number of susceptibles, the number of resistant individuals is lower in the population compared to the high selection, but also there's less of a chance that these two resistant individuals will mate, so that we know their offspring will be resistant, and it increases the chance that, say, a susceptible and a resistance will mate, and thus their offspring will have a mix of susceptibility and resistance and may actually die after exposure to a pesticide. So for example, if we've got a field with a mix of resistant and susceptible individuals, we spray it and kill off all, or at least the majority of susceptibles, then the only individuals we have left at mating are our two resistant ones. And if they mate, we know that all their offspring are going to carry resistance traits, and we're going to be out of luck. On the flip side, if we were to, say, have some sort of area where there were susceptibles left over, or if we applied a spray that was less selective that saved some susceptibles, there would always be the possible that those susceptible individuals would mate and mix their genes. So, how do we reduce selective pressure? Well, really, the number one way to do this is just to apply less pesticides, right? If we apply fewer pesticides, there's less selective pressure, less mortality occurring, and thus less selection favoring resistance. And so there are a couple ways that we can get about this. One of the most commonly used ways is the use of economic thresholds. So essentially, if you only spray when you know you're about to have economic damage occur, then you're going to reduce your total number of sprays. In general, because you're not just spraying every time you see a bug or on a calendar, you're only treating when you need to. On the flip side, you can use basic IPM. You can integrate multiple control techniques. And the idea here is that if you have cultural, mechanical, biological, and behavioral control all working together, then you're generally going to have more help keeping the pests below that threshold, and thus you're going to need fewer sprays, and thus have lower selection pressure. And then finally, again, sort of the standard IPM solution to pesticides, which is, if you're going to use a pesticide, use a selective application as much as possible. That'll protect your biocontrol agents, and it's going to increase the amount of control that you have of the remaining pest population. And again, decrease the chances that you'll have to apply one more time. All right, so let's say though that you've got a pest that you are regularly applying pesticides to. This is the reality of a lot of pest systems right now. Things like the California red scale in citrus. There is some biological control, there are some cultural approaches, but for the most part, if you're not spraying at least once a season, then you're going to get pretty big populations and you're going to take a hit when you go to the pack house. So what do you do when you have to apply? Well, what you can do is you can reduce the selection pressure that favors a single resistance trait by rotating your modes of action between your applications. So essentially the idea behind this is that if you're changing your mode of action, you still have a lot of selection pressure favoring resistance, but you're going to be targeting and favoring different resistance genes every time you spray. So generally, a resistance gene is going to most commonly provide resistance to a single mode of action. So if you're an insect and you have a resistance gene that provides resistance to Bt, it's not going to help you if you get exposed to an organophosphate or a pyrethroid, or a sodium gate or a neonicotinoid or whatever, right? It only works on Bt. So if you spray Bt, you're going to favor Bt resistance. The next generation, you're going to have more Bt resistant caterpillars. But then if you spray that next generation with a neonic, then those Bt resistant guys aren't going to be favored at all. They're going to die out, right? And then if the next time you spray a pyrethroid, that's not going to favor Bt or neonics. So generally, the more modes of action you have available, the more widely you rotate them and space out those rotations, then the less selection pressure there is, favoring a single trait. Plus, because those resistance genes have a fitness cost like we talked about last week, when we reduce that pressure, they're actually going to decrease in frequency. because the susceptible individuals that don't have that trait are going to outcompete them, and they're going to mate more successfully without that pesticide around. So this is all basically posited in the idea that resistance genes are going to be rare, that it's really unlikely that a pest is going to have more than one mode of action, more than one resistance gene for more than one mode of action. So it's unlikely that there's some pest out there with resistance to Bt and pyrethroids. And thus by rotating, you're able to kill pests that are resistant to just one MOA. Now, admittedly, mode of action rotation is not going to work as well if you're targeting a pest that is resistant to multiple pesticides. So something like a cross-resistant pest. You're not going to get a whole lot of benefit out of rotating between the two pesticides that it is cross-resistance to. On the flip side, when you do a rotation, you can't include a pesticide that you already have resistance for in your population. So let's say you've been spraying with mode of action W for a while now, and you're starting to see resistance. By switching over to a rotation using mode of action W and mode of action X, what you're really doing is you're selecting for the individuals that are resistant to mode of action W and resistant to mode of action X. And so you're going to create multiple resistance. Generally, when you're rotating, you're going to want to have new modes of action for more or less every single step. Things that the pests are unlikely to have been exposed to in the past. So now this may raise a question that you're asking, well, if it's hard for pests to develop a resistance to, say, two different pesticides, say a neonicotinoid and an organophosphate, then why don't we just apply two pesticides at the same time? Why don't we do a tank mix of a neonic and an organophosphate? That way we're exposing the pests to two modes of action at the same time, and it's highly unlikely that they're going to have resistance to both, and we're going to kill them off. Now, in theory, this is a really good idea. It would work in a large part. However, the challenge here is that, for the most part, pesticides are all put through environmental trials alone, right, as single chemicals. And so it's not really well known how they interact when they're mixed, whether they actually work the same as two independent factors, or maybe they synergize. Maybe they actually interact together to kill insects more effectively. Or on a third alternative, it's possible that they get in each other's way, so that they don't work as effectively as mixed. But the thing is, what we found through a couple of studies is that mixtures of pesticides tend to have lots of unintentional environmental effects, especially on non-target organisms. So, for example, this was a study that was done in the Pacific Northwest, looking at the effect of acetylcholinesterase inhibitors on salmon populations. So acetylcholinesterase is an enzyme that is essential to the functioning of the nervous system. It's used to, acetylcholinesterase inhibitors are used to control insects, but they do have some toxicity to mammals, fish, lizards, anything with a nervous system, because we all use acetylcholinesterase. So, typically though, they're designed in such a way that they will target the enzymes in insects while leaving, say, fish unimpacted. And so there's a variety of acetylcholinesterase inhibitors that are approved for application in watersheds with salmon because they're known to have very low toxicity in the watershed. But a team of researchers recently looked at, well, what happens if we mix two different types of acetylcholinesterase inhibitors, carbamates and organophosphates, and we start, say, mixing them in the tank and seeing what their impact is for toxicity to these fish. So what we have here on this graph is different mixtures, such as a control plus I believe this is a carbamate, and then we have various mixtures of carbamates with organophosphates or organophosphates with other organophosphates. And then on the y-axis here, we have the percent activity in the fish of their acetylcholinesterase. So the idea here is that the closer to 50% you are, the healthier the fish is. The closer to 0% activity you have, the more sick the fish is, the more danger it is of dying. So what we see is in the controls, we have a little bit of inhibition, about 12–15% inhibition. So, these pesticides are having an impact on the fish. But what we see is when we start mixing chemicals, some of them are fine, they don't really hurt the fish, but then you start getting into treatments where you're doubling or even tripling the amount of acetylcholinesterase that's being shut off in that fish species, so that the toxicity increases synergistically when we mix our pesticides. Consequently, without knowing how pesticides interact with one another, it's not safe to go out and spray them. There can be all sorts of unintended environmental consequences, and so if it doesn't say it's safe on the label, then don't mix. Don't bother with that. Finally, we can also use something called a synergist. So a synergist is a chemical that we can add to our pesticide mix that will inhibit the pest detoxification pathway. So these are sometimes used against insects because we have things, specific chemicals that are capable of inhibiting oxidase inhibitors, or cytochrome P450s. Those enzymes that insects typically use to detoxify pesticides. And if we mix those inhibitors with our pesticide, we basically shut down the insect's detoxification pathway. And so an insect that is naturally resistant will be unable to resist the pesticide. And by doing this, we essentially can block or at least slow down the development of resistance, because those individuals that are resistant aren't going to be favored near as much as they would be otherwise. Now, this isn't used very often because it oftentimes is much more expensive than just applying a new pesticide. However, a lot of companies are investigating RNAi-based technologies that could be an inexpensive genetic approach to synergizing pesticide effects. And so that might be something to keep an eye out for in the future as a way to limit detoxification. All right. So, finally, my mistake. I thought we were moving to the next section. So finally, I believe the last way that we can really reduce selection pressure is just to use less persistent pesticides. So generally, the longer the pesticide is in the field, the longer the pest is exposed to it, and the more of them are going to die just due to exposure. But if you use a less persistent one, you actually increase the number of susceptibles that can move into the field after the treatment and increase the chances of crossbreeding between susceptibles and resistance, basically producing hybrid offspring that are more likely to be susceptible than a resistant versus resistant mating. Similarly, as we mentioned before, you can avoid spraying directly before mating to have the same effect, give the pests a little bit of time to reestablish. And also, you can treat locally rather than treating the whole area. And as we mentioned earlier, the real advantage to this is that if you target the areas that are hot spots for pests, then you'll leave certain areas that act as a refuge for your susceptibles, and they can move in and mate with those resistant pests. So for example, if you've got, say, a set of fields like this all right next to each other, and you notice you've got a pest outbreak in your middle one, it might be tempting just to apply pesticide across all three. Because if you're using something like an aerial application, you might be sinking more costs into hiring, say, an airplane than you are in the pesticides. So you might just say, you're already flying over all three fields, just spray all three of them and take care of it. However, if you were to just spray the one field, you'll at least maintain some populations of susceptibles in your neighboring ones that can go in and mate. All right. So the nice thing about sort of this targeted approach here with reducing selection pressure is that it segues really nicely into the next way that we can control resistance, which is by increasing the number of susceptibles. They're really more or less the same idea, but just slightly different approaches to it. So again, the basic idea is that if we preserve susceptibles, then they are more likely to mate with the resistant individual than a resistant individual is to mate with another resistant one. And this is just because resistant individuals are relatively rare, so it's hard for them to find one another. So if we were to flood the landscape with a ton of susceptibles, then it's more likely they will mate with each other and produce some sort of hybrid offspring that may be susceptible as well. So one way to increase the number of susceptibles is to create refuges for these susceptible pests. Or in other words, have a part of your field that is more or less just left untreated when you are out there applying the pesticides. This will act as a safe haven that the susceptible pests will live in, and then when the mating period comes, they will be available to go into the neighboring sections and mate with those resistant individuals. Ideally, these hybrids will be susceptible to the pesticide, and that way when you spray, you're eliminating not just a pest, but also some resistance genes that may be masked by the hybridization. Now, this approach was used very successfully recently with the introduction of GM corn and cotton. So back in '96, genetically modified cotton and corn that expressed the Bt toxin were introduced to the United States. Now, they're not really widely used here in California, but they are very popular in the Midwest and the Southeast, where there are lots of lepidopteran and coleopteran pests that get after corn and cotton. And now when they introduced them, there was a lot of concern in the entomological community that their utility would be really short, because these are basically like putting out a plant that is constantly coated in an insecticide, right? It's got ultra-high selection pressure. They designed this thing so it expressed so much Bt toxin that more or less it killed 100% of all the pests that feed on it that aren't resistant. So you lose all of your susceptibles, and because it's always toxic, it doesn't matter when the pest shows up, it's going to die. So it's not like you can spray at the beginning of the season, to avoid spraying right before mating. They're always going to be exposed before mating. And so there was a team that was assembled, about maybe 30 or 40 entomologists to study this problem, and they created a model that predicted that if this was widely planted across the United States, there would be full resistance in all pests targeted within 3 growing seasons. So essentially, it would be a next to useless technology. So they came up with a solution, and they got it passed through the US EPA, that they mandated that 20% of the field would be planted as a refuge, which is now closer to 5%, but the technique is still used. So essentially, 1 out of every 5 plants you planted had to be not Bt. It had to be some sort of susceptible corn or cotton variety. And it didn't really matter how you planted it, whether it was a linear block, borders, strips, whatever, just so long as you had it directly next to your field of Bt. And this actually worked out to be really, really successful. Using this approach, resistance was delayed to the extent that the first case of resistance in the United States was found 15 years after the introduction of GM, cotton, and corn. So that would be about 2011. So they got basically 5-fold more growing seasons out of this than they would have otherwise. And that resistance is highly localized. So there are hotspots of resistance. Generally, the problem they found with resistance is that they are in areas where growers were planting what we call constant corn or continuous corn. So they weren't rotating their corn with soybeans or any other crops. So they were constantly having the same pests over and over again. There's also some questions that in those areas, growers might not have been planting refuge. They might have just been taking advantage of the fact that their neighbors were planting refuge, and so they were kind of cheating the system. And to that extent, the vast majority of pests across the United States are still susceptible to Bt to this point. And so these refuges appear to be working very well. What's funny is that there was an area, Puerto Rico and parts of Latin America, they found that they had resistance within about 3 growing seasons. And the main reason for this was that, again, a lot of growers weren't following the recommendations for a refuge, but also they get a lot more growing seasons in these tropical areas than we do in North America. So whereas we may be getting 1 corn harvest every year, in parts of Puerto Rico, they may be getting 2 or maybe even 3. So over 3 growing seasons, it's more like 9 pest seasons for them. And so the refuge helped them out a little bit, but again, it's not going to help you if most people aren't doing it. Nowadays, what they do is, because they're this 5% refuge, the BT, the GMO makers, will actually just mix the susceptible plants in with the resistant seed so that it gets planted randomly throughout the field. All right. So, there are challenges to resistance management. And I guess that should be expected. I mean, if people aren't doing it, something that's as clearly helpful as resistance management is, it means that there are certain problems. So one of the big challenges is the one that Dave talked to us in lab about, which is that it's really difficult to manage resistance if you don't have very many modes of action. So insects tend to evolve resistance very quickly, and so MOAs tend to rotate pretty quick. You know, you have an MOA that works one year, and maybe 6 seasons later, it's not any good. And so it's very hard to develop a good rotation scheme to control your resistance. Whereas on the flip side, weeds and fungi seem to evolve resistance much slower than insects, so they have lots of modes of action, and you can rotate them, and by doing so, you actually increase the lifespan of these technologies. And so it's just sort of a nice positive reaction loop, right? Because they evolve slowly, we can rotate, which allows us to make them evolve even more slowly. And it's why in weeds and fungi, the big problems you see tend to be in fungi like gosh, I forgot the name. Well, let's skip to weeds. That's why in weeds, you tend to see the most problems in systems where you're using the same pesticide over and over again, like glyphosate, and in fungi when you're dealing with fungi that have very few MOAs available to them. Gosh. Well, I can't remember the name of the fungi I was going to talk about, so I'm sure it'll come to me later and I'll mention it then. So another challenge is that resistance management can be really tricky in specialty crops. And I talked about this a little bit earlier, but it's the basic idea that in a specialty crop, consumers don't want any pest damage. You don't want to run the chance that you're going to pick a blueberry out of a bag and bite into it and get a mouthful of spotted-winged Drosophila larvae, right? And so, essentially, there's a really big trade-off for the amount of damage and the amount of profit the grower gets. That if you have more or less any damage, you're going to get bumped down a quality standard. And you're going to get less money for everything you produce. So consequently, PCAs and growers are pressured very heavily to use pesticides not only frequently, but also to use the pesticides that have the very strongest selection pressure, the highest mortality. So basically they're creating sort of a perfect storm. You apply a ton of pesticides and you apply very strong ones. And then finally, there are knowledge and cost constraints. Pesticides differ in price, application technique, their effectiveness, their selectiveness, all of these things. And it takes a while to learn all of that information. And so it can be really difficult to integrate all of them effectively so that you maximize the control that you get. And as such, this can turn people off, especially if you're a busy PCA who's managing a lot of fields. You may not have the time to really invest in developing a good resistance management scheme. So one of the ways they try to get around this is that oftentimes a label will have a resistance recommendation section on it where they'll tell you when to apply this and how to rotate it. And that might be an easy way to sort of circumnavigate a lot of these knowledge and cost gaps. So with that, I am finished. I will see all of you on Thursday where we will be talking about pesticide application safety and health concerns. All right, see you then.